So let me start with a quick overview. Last week we started to talk about underdetermined systems in metabolic network modeling, mass balance-based modeling. So these are the systems with degrees of freedom greater than 0, and three major constraints are important. Equality or mass balance-based constraints. This is S dot V equals 0. Reversibility constraints. So this is based on if the reaction is reversible or not. For reversible reactions, the reaction rate can take any value between -infinity and +infinity. For irreversible reactions, the rate has to be positive. And the third constraint is the measurement constraints. So we measure some rates. This is also important to decrease the degrees of freedom or to make it equal to 0. So we use those measured rates as inputs. One issue maybe I didn't talk about last week. So we have this reaction. If the reaction rate is 3, the measured rate is 3. So we use this style to define the reaction rate. In this case, the minimum value of the reaction is 3, the maximum value of the reaction is 3. So we constrain the reaction rate to be exactly equal to 0, the measured rate. What does it mean if the reaction rate is negative? So negative reaction rate means the reaction proceeds in reverse direction. So from C to D, the rate is -5. So this means that the reaction is actually proceeding from D to Z, D to C with a rate of 5. So there are those major constraints to solve the related underdetermined system. This approach is also known as constraint-based analysis. And flux balance analysis is the most common constraint-based analysis method. It uses optimization to calculate unknown fluxes. And optimization requires an objective function. So we talked about this very simple FBA demonstration last week. We talked about the first FBA example in literature, 1986. And we talked about the major optimization types. The optimization we will use for the solution of underdetermined systems is of convex type, and mostly we will use linear programming or quadratic programming. Linear programming means the objective function the function that we want to minimize or maximize is linearly combined. So we don't have any quadratic terms, any terms with a power of 3, 5, etc. For quadratic programming, we have a function to maximize or minimize, and the terms are either linear with a power of 1 or quadratic, so to the power of 2. So how to choose the objective function? It is important that the objective function we will choose must be biologically meaningful. It must be in accordance with the phenotypic behavior of the organism. So the most commonly used objective function, therefore, is biomass reaction maximization for microorganisms. Microorganisms, they keep it is based on the cell division, right? And the rate of growth or the production of biomass is at maximum. This is the assumption. So microorganisms try to grow as much as possible. This objective function was in use in 90s, and still it is the most popular objective function used in constraint-based modeling of metabolic networks. And there was a paper published in Nature in 2002 which in a way proved that the growth maximization is a relevant objective function for microorganisms. So this was the framework for underdetermined systems. I said that most of the time we will use linear programming to solve underdetermined systems. And in MATLAB, there is a function called linprog for linear programming. This is the function, and it solves a problem which defines equality and non-equality constraints, inequality constraints. So this part is the same as our s.v equals 0. And we don't have any A or B because we don't have any inequality mass balance or something. And our AEQ matrix will have a dimension of m by n, m mass balances, and unknown reaction rates. X is our vector of unknowns, rates, and BEQ is a vector of zeros with a dimension of m by 1. LB and UB here are the lower bound and upper bound vectors. So for such a system, for example, let's say we have a system with 4 rates, and the fourth one is measured, so it is constrained with the measured rate in the lower and upper bound. This is a reversible reaction, this is a reversible reaction, this is an irreversible reaction. In this case, our lower bound vector will be this part, and our upper bound vector will be this part. And so the dimension of lower bound and upper bound vectors is the same as the dimension of unknown vector, V. And for F, let's say if the objective function for this system is maximizing V3, so this is how we represent F. Only V3 is available in the objective function. So for V1 we will enter 0, for V2 we will enter 0, V1, V2 and V4 also. They are not available in the function that needs to be maximized. And we have V3, for V3 we will have 1. One important point, in linprog and in many other linear programming solvers, if you want to maximize the objective function, you multiply f by -1. If you want to minimize the objective function, you multiply it with +1. So that's why we have this minus here. Optimization means either minimization or maximization. So this is how the solvers implemented these two alternatives. If you multiply the function to be maximized, in this case represented as f, by -1, this means that you maximize that objective function. Before proceeding, I would like to share with you a graphical example for linear programming. A very simple example of two unknowns. And these are the constraints given. V1 is constrained to be between 0 and 60. V2 is constrained to be between 0 and 50. And V1 and V2, V1 plus 2V2 must be smaller than 120. And the question is, what is the maximum value of this? So this is our objective function, the function to be maximized. So what is the maximum value of this function under those constraints? And the example is figure 18.5 from Bernard Paulson's book, Systems Biology, Constraint-based Reconstruction and Analysis. So this is a graphical example. So we will solve this problem by using a two-dimensional coordinate system. And this way you will better understand how the linear programming and linear programming based optimization works. So the strategy is we have two unknowns. We can represent those unknowns as two coordinate systems. So V1 represents x-axis, V2 represents y-axis. So a graphical solution on 2D, two-dimensional coordinates is possible. So this was our first constraint. V1 must be between 0 and 60. This is V1 axis. This is 0. This is 60. So V1 can have values anywhere here, right? It cannot take any values outside this boundary. What about V2? V2 can have values between 0 and 50. So this area. Right? It is bounded by this shape. So V2 can take any values between here, not here. So in this coordinate system, the area that are both satisfied with V1 and V2 constraints are this area, right? Here, V1 is not valid. Here, V2 is not valid. So in this area, both V1 and V2 constraints are true. So this is our current solution space. With those constraints, we don't have a single solution, but we have a solution space. So any point in this solution space holds our constraints. We have one more constraint. V1 and V2, V1 plus 2V2 must be smaller than 120. This is high school information actually. So if you have something like this, we know that this corresponds to a line in a two-dimensional system, right? And if it is an inequality, it defines one side of this line defined with the equation. So the thing we need to do first is to draw the line defined by V1 plus 2V2 equals 120. So in order to easily draw this line, we can assign 0 to V1 and calculate the value of V2, which is 60. So this line will pass from V1 0, V2 60 points, so from somewhere here. And then let's assign 0 to V2. In this case, V1 takes the value of 120, so here somewhere V2 is 0 and V1 is 120. So we have found 2 points on this line, right? So the line will pass through these two lines. So this is the line of V1 plus 2V2 equals 120. And in our case, it is inequality constraint and it is smaller than. So this area is the area that corresponds to the given inequality constraint. And again, in this graph, we need to find the area that are satisfied by all three constraints. So if you look at here, this is the area that is satisfied by all three constraints. This is our solution space, constrained by the given three constraints. So the next step is implementing the objective function. Our solution space is defined like this, and in this solution space, we want to identify the maximum value of a function given as 20v1 plus 30v2. So what is the maximum value that this function can get within this solution space? So we want to find the value that this, let's call it as z, but we don't know it. The maximum value that this function can get within this solution space, let's call it as z, but we don't know its value. That's the question. What is the value of z given this solution space? At this point, let me remind you how we can easily calculate the slope of a line. For this line, for example, if we are asked what is the slope is the deviation of the y-axis, in this case represented by v2, over x-axis divided by y-axis. So then the slope of this line is 1, because 1 is the coefficient of v1, the x-axis representation. So the slope of the line is 1 over 2. If our objective function is defined like this, and we don't know the value of z, this is also a line, right? And the slope of this line will be 20 over 30. So with the given objective function, we don't know its value, but we know that it represents a line, and we know the slope of this line. So, for example, this was the line of v1 plus 2v2 equals 120. If I draw another line which is parallel to this blue line, it will have exactly the same slope. So it will again be defined by v1 plus 2v2, but this part will change. So what is v1 and v1 plus 2v2? What is the value of v1 plus 2v2 for this line? We don't know, but it has exactly the same slope. You can calculate it because we know that this line passes through a value of V1 50. And in this point, it also passes through a point with V1 60 and V2 certain value. So I won't go into details, but yeah, with a simple calculation, you will see that this line is equal to 100. V1 plus 2V2 equals 100. So these are two lines with the same slopes, but their values are different. So what does this mean? Here, in this solution space, we are searching for a line with a slope of 2/3. So this is 60, this is 90. If you calculate the slope, 60/90, it is 2/3. But we don't know where exactly this line is. Should it be, this is also a line parallel to the same line, the green line. This is also parallel to green line. This is also parallel to green line. This is also parallel to green line. So which of those lines are maximizing the solution space? Which of them have which of them is still satisfying the constraints defined by the solution space and has a maximum value? This line, for example, it passes from point 60, and this is its value. So the value let's take this point, for example. This is the formula. In this point, V1 is 60, V2 is 0. So if you calculate it, 20 times 60 plus 0, which makes 1,800. So by using a single point on the line, we can calculate this value, because that value will hold in any point on the line. So we want a line which is parallel to this green line and still lie on the solution space but has a maximum value. And we know that this value gets higher if you go upper, in an upper place. So this means that this will be the line which is still in our solution space and has a maximum value. And if we check we will see that this value is 2,100. So this was the solution. This was the original question. Given the constraints, what is the maximum value of this function? So this is the objective function. And by using a graphical approach, we were able to solve, first define the solution space, and then find the point within the solution space that maximizes the value of the given objective function. So I shared this with you to give you an idea on how linear programming works, how the constraints and the defined objective function are used together to get a solution. So our constraints define a solution space. Here you see it's in three-dimensional space. From one of the key papers on flux balance analysis published in 1994. So our optimum point will be one lying on the corners of this solution space, because we want to maximize the value of an objective function. So then our objective function will not lie inside the space, but it will be on the corners, on the outer edges. So another figure that tells something similar, our constraints define a space, right? If we have n unknowns, it will be an n-dimensional space. And within this space, we will have a shape that defines the solution space. And if you want to maximize an objective function, this will correspond to a point on the edges of this solution space. So it can also be here. This is also the edge. Or this is also an edge, right? Because this is also an, yeah. So when we use biomass as the objective function to maximize the biomass reaction rate, what does it mean? The cell takes glucose from outer environment and uses it via glycolysis or pentose phosphate pathway and TCA cycle, right? And produces building blocks which are later used to make macromolecules. So when we maximize biomass as objective function, we maximize the flow of carbon from glucose to the macromolecules, lipids, proteins, and nucleic acids. We tell that all the glucose must go to these macromolecules within the allowed constraints. So we maximize the flow to synthesize biomass precursors and also building blocks and also macromolecules. So synthesis of biomass precursors, metabolic precursors, are maximized. So these are roads, for example, that goes to macromolecules from metabolic precursors. So we maximize the flow of carbon through those roads. Before switching to a class exercise, you know, there isn't a, in my opinion, there isn't a good book that would be used as a textbook when it comes to the metabolic network modeling. Instead, it is more useful to read review papers published on the issue. So I would strongly recommend you to read those papers. The first one was published in 1994. It was the first review on flux balance analysis. About 10 years later, another paper was published on advances in flux balance analysis. And in 2010, again, Paulson's group published a paper on definition of flux balance analysis. which was cited almost 3,000 times. So if you want to learn more, rather than trying to search